Hello, my name is Cecia Alvarez, and today I'm going to talk about the hypothalamic arcuate nucleus, a key site for mediating leptin's effect on glucose homeostasis and locomotor activity. So obesity and diabetes are some of the most detrimental, common, and increasing diseases around the world. Understanding the cellular background and signaling pathways behind these conditions is key when it comes to the development of preventive and treatment mechanisms. Leptin is a hormone that plays key regulatory roles in body weight, glucose homeostasis, and locomotor activity. Investigating leptin's effect and signaling pathways could bring some light into new therapeutic options for several illnesses, as well as motivating further in-depth investigations surrounding this multifunctional hormone. So leptin is a hormone secreted by the adipocytes and communicates to the brain the energy status of the body. Also, it has been shown its key role in different regulatory homeostatic pathways involving glucose, locomotor activity, energy usage, obesity, food consumption, neuronal plasticity, and possibly infertility. Leptin's primary site of action is the central nervous system, where its receptors are found in several brain regions. However, the hypothalamic arcuate nucleus has shown to be an important activation site for leptin. So for this experiment, we are going to focus on mice LEP-R-Neo, that is a mice homozygous for FLPE reactivatable leptin receptor null allele. The researchers had other mice populations, such as mice LEP-OB and mice LEP-RDB, one lacking leptin and the other one lacking receptor signaling for leptin. And both of them were obese, diabetic, infertile, and hypoactive. So we can already see a correlation in between leptin and these negative effects. So the purpose of the experimentation was to restore leptin signaling in the arcuate nucleus of the LEP-R-Neo mice. This was performed by producing an adeno-associated virus expressing a FLPE recombinase vector, and this viral vector that would also express EGFP for visualization purposes was stereotaxically, meaning was manipulated directly into the brain through injections into the hypothalamus, focusing on the arcuate nucleus of LEP-R-Neo mice. Due to the complexity of the procedure, some injections were a hit and some were a miss in respect with reaching the arcuate nucleus. Thus, mice populations were further divided into hit LEP-R-Neo mice and miss LEP-R-Neo mice, and the miss injections were considered as a control group. So if we can see here in table 1, it's showing after the injection, the hypothalamic distribution of the induced leptin in the neurons. And it's clearly the difference in between both the populations receiving the HEAT injection and the miss injection. And in figure 2, after the injection, the HEAT LEP-R neo mice were showing higher immunoreactivity in this region. So again, the approach of this experiment was to deliver this viral vector in LEP-R-Neo mice to allow the researchers to express these endogenous leptin receptors in these neurons. And the results were as follows. So after 7 weeks, the body weight started to decrease. Fat mass and food intake also decreased, and the energy usage did not improve. And this is comparing the LEP-R-Neo mice with the HEAT and the miss injections. Also, glucose homeostasis improved significantly in between 4 to 8 weeks after the injection. Reproduction in both groups did not improve in either in HEAT or in miss injections because they were unable to produce offspring. And locomotor activity significantly improved after 24 hours of receiving the injection. Here are the graphs for the results previously explained. And in conclusion, leptin is a hormone released by adipocytes that plays a critical role in homeostatic roles in different pathways. Leptin's primary site of action is the central nervous system, and its proposed primary activation site is the arcuate nucleus in the hypothalamus. Glucose, fat mass, locomotor activity, food intake, fertility, and body weight are some of the systems in which leptin signaling could and plays a fundamental homeostatic biological role. And through the analysis of the findings after using an adeno-associated viral vector expressing FLP recombinase stereotaxically injected into the arcuate nucleus of the hypothalamus in these LEP-R-Neo mice population, it was found that and reinforced leptin signaling homeostatic role in almost all the systems previously mentioned. Here is my bibliography, and thank you for your attention.